Okay, everybody. Hopefully you got out and got a chance to enjoy the weather the last couple days. It's been really nice out. I just wanted to review quickly the three tools that we've touched on so far in biotechnology, sort of the more advanced form of genetic engineering, before we move on to the last topic. And so, just a quick review here then. What are we looking at? So we've got BAM, and Hindi, and EcoR1. And if you take a look, we've got small sections of DNA. Obviously, the DNA extends off this way and off this way. So let's just assume this is a gene or a piece of DNA. And if you take a look, all of these have sort of different sequences that they look for to cut. So this dotted line would be cutting between the hydrogen bonds, between the nucleotides, and then also would be cutting sort of here and here. So hopefully you remember or recall and know what we're looking at here. These are restriction enzymes. And the difference between them is, number one, they come from different bacteria. And number two, they cut at different sequences. So you can put one in with some DNA, you can put two, you can put all three in. And the more restriction enzymes you put in with your sample of DNA, the more it'll get cut up into pieces. Okay, now remember, just because you cut your DNA up, that doesn't mean you have enough to work with. One little piece of DNA is very hard to detect, but if you take that one piece of DNA, or many small pieces of DNA, and you run them through PCR, right? Polymerase chain reaction. Remember, that's an enzyme and some heat and cooling to take one piece of DNA and turn it into many. You have something to work with, okay? So, what is that you can work with now? Well, hopefully you got something out of the virtual lab the other day, but it's this. This is sort of a representation of gel electrophoresis. So if you'd imagine we're looking down on a gel here, and up on top we have wells, well number 1, 2, 3, and 4. And if we were to put DNA samples in here, along with restriction enzymes to cut those samples up, right, in each of these wells, and they might be different samples and different restriction enzymes, it's hard to say for sure. And then we were to run that gel. Remember, DNA has a negative charge, and so it's going to tend to migrate out of the well towards the positive end of the gel over time. If you just let this run, all these bands of DNA would just run right off the end and out of the gel. I've seen that happen. So let's see if we can determine what is this telling us. So first of all, are any of these samples from the same organism? So let's just assume that we're using the same restriction enzymes, and we've put some different DNA samples in each of these, but we're using the same restriction enzymes, so they should be getting cut in the same spots. So are any of these samples, 1, 2, 3, and 4, from the same organism? How would we know that? Okay, well, yes, lane 1 and lane 4 are. How do we know that? Look at the banding patterns, right? It's like a fingerprint. So this line here matches this line here. This line here matches this line here, right? So if you look at the pattern down this column versus the pattern down this column, this tells you that this in fact is the same DNA, because if we were to cut it up and it ends up with the same lengths coming out of that well, then we're looking at the same DNA. Remember, yes, DNA has that negative charge. Okay, and then also another question you might need to be able to answer about gel electrophoresis is, if the DNA traveled way out here, is that a shorter or longer piece of DNA compared to up here? Hopefully, you recognize that these are the shorter pieces. So the long pieces of DNA have a hard time moving out of the wells. They don't move very far over the same amount of time, but the short pieces tend to move faster through the gel, and they make it further in the same amount of time. So this is a way to spread out your DNA. Now, we don't necessarily know what any of this DNA does yet. But we could literally take a scalpel and cut out a sample of this DNA, right? And from there, we could try and recombine that DNA with a viable piece of DNA, with a piece of DNA that would have access to a ribosome and the ability to make a protein. So that we could determine, okay, does that small piece of DNA have genes in it that could make proteins? And how would we know? Well, if we take a bacteria and we already know what that bacteria makes for proteins, and we put a piece of DNA that we just took out of that gel into this bacterial DNA, and it produces proteins that we've never seen before, well, then we know that we've added some new genes to it. So how do you do this? So now, basically, right, here's a human cell. We take the DNA out of that cell. That's another lab we would have done in class, right? A DNA extraction, fairly easy. And we cut that genetic material up. Remember, we're talking about chromosomes. And now we take some of that genetic information, hopefully it has some genes in it, and we recombine it with DNA from a bacteria. Well, how do we do that? Well, guess what? We use the same restriction enzyme to cut into this bacterial plasmid, which is just a circular piece of DNA inside a bacteria, as we did to cut into the chromosome. And because we did that, we'll have the same ends, right? The same sticky ends. And therefore, it'll be easy to splice in this piece of DNA into our bacterial DNA, and then we can put that plasmid back into the bacteria. If in fact our DNA made it into the bacteria and we let it reproduce for a while, eventually that bacteria will be producing what that gene tells it to produce, the human gene tells it to produce. And the first case that this happened was in the production of insulin. Okay, so insulin is, of course, a human protein, right? A human gene says how to make that protein. And I think it's on chromosome 11, if I remember correctly, if you're thinking of a karyotype. And researchers cut it out of a human cell using a restriction enzyme. They spliced it into a bacterial cell using the same restriction enzyme. They allowed that bacteria to reproduce over and over again. And guess what? That bacteria, instead of not only made its own proteins, it also started making the human protein insulin, and lots of it. In fact, that's how human insulin is made now, compared to how we used to be able to get it out of the islet cells or the pancreas of slaughtered animals. Right now, it's coming from a bacterium made using the ribosomes of a bacteria. Okay. So again, what do we normally use to do this process of creating transgenic organism? There's a term we're going to be using moving forward, trans across genic, pertaining to genes, right? So in other words, genes moving across from one organism into another. Bacteria were the first transgenic organisms, right? Because it was fairly easy to manipulate them, fairly easy to sneak other DNA into their cells, okay? So plasmids are generally what are used because they're easy little pieces of DNA to manipulate. And that's what we're going to be talking about moving forward, recombinant genetics. So I also have a Khan Academy for you to watch. He explains recombinant DNA, maybe again for you. So hopefully between the two of them, it starts to click for you, starts to make sense. And then don't forget to take your daily evaluation. Only one more day this week. Remember, we're off next Monday, so we got a 3-day weekend coming. So stick with it. Take care. I hope you're all well.